Hello everyone, good evening to all. Welcome to One Shot NEET 2025 Rapid Revision Program. So this is the second video on reproduction, human reproduction and reproductive health. In the previous video, we had discussed about the human reproduction up to the spermatogenesis only we had completed. Now in this video, we completing the remaining topics related to human reproduction and human reproductive health. So these chapters are most weighted chapters in NEET examination, right? So there is a chance to get more questions from these chapters. That's why we are only focusing on the most weighted chapters for this 15 days up to 28 April, right? So now let us start the second video on human reproduction and reproductive health. So that is oogenesis is our the next topic. Oogenesis. Oogenesis. So you know that oogenesis, production of the female gamete, production of the mature female gamete from the primary sex organ, that is ovary, is called oogenesis. Okay, production of or formation, production of mature female gamete that is ova is called oogenesis, right? Oogenesis is the process which is initiated during the early embryonic stage only, right? When female fetus developing in mother's uterine cavity, so that time only oogenesis process is initiated. The oogenesis process initiated during the embryonic stage in the uterus of mothers, right? Now during the embryonic stage, during the embryonic stage, during the embryonic stage, in each fetal ovary, each fetal ovary, in each fetal ovary, 2 millions of gametes, mother cells, 2 millions of gamete mother cells, gamete mother cells that is oogonia will form. Okay, suppose imagine, so this is the fetus, inside the fetus you can see these are ovaries, inside the fetus these are ovaries. Okay, so when these ovaries are developing during the embryonic stage, within these ovaries couple of millions of gamete mother cells, couple of millions of gamete mother, couple means two, that means 2 millions of gamete mother cells, that is oogonia, present in each ovary. Each ovary contain 2 millions of gamete mother cells oogonia. That means total number of gamete mother cells present in ovaries, total 4 millions. Very, very important. 4 millions. Now, these oogonia never be formed or never be added after birth. Okay, their number will be only developing during the embryonic stage. After the birth, they never be formed and they never be added. Okay, so this is very, very important, right? Now these oogonia later they will modify into primary oocyte. Primary oocyte. Okay. Primary oocyte. Oogonia will modify into primary oocyte. Suppose this is oogonia. The content of the oogonia or the DNA content of the oogonia will increase and it will modify into primary oocyte. This is called primary oocyte, right? Primary oocyte. So now in this diagram you can see this is the primary oocyte. Primary oocyte. The primary oocyte surrounded by one layer of granulosa cells or single layer of granulosa cells. These are granulosa cells. Granulosa cells. These granulosa cells are present in stromal tissue of the ovary. So ovary inside peripheral region, outside central regions, that is we know that, right? So granulosa cells are present in ovary. That granulosa cells are surrounding the primary oocyte. Okay, inside this is primary oocyte. Primary oocyte. Primary oocyte. Okay, so primary oocyte. When primary oocyte surrounded with single layer of granulosa cell, now we are calling it primary follicle. We are calling it primary follicle. So this is very important question. How can you identify a primary follicle in female? Okay, so primary follicle, very important, right? When oogonia, this is oogonia developing into primary oocyte. Primary oocyte, right? Now these primary oocyte undergoing meiotic division 1. Meiotic division 1. It is started, meiotic division started in primary oocyte, and that meiotic division is arrested in prophase 1 of meiotic division 1. Okay, from the oogonia, primary oocyte is formed. Now that primary oocyte undergoing meiotic division 1 and that meiotic division is arrested in prophase 1. Prophase 1. Now you can see this is the primary oocyte. It was arrested in prophase 1 meiotic division 1. Prophase 1 meiotic division 1. Now these primary oocyte surrounded by single layer of granulosa cell. When primary oocyte surrounded by single layer of granulosa cell, we are calling it primary follicle, right? Soon after, this primary follicle transform into secondary follicle, right? So I will take this diagram, you can see here. This is the primary oocyte. Now this primary oocyte surrounded by more layer of granulosa cells. You can see here more layer of granulosa cells are there. Earlier there was only single layer of granulosa cells, but you can see the granulosa cell layers increased. The granulosa cells are increased. More layer of granulosa cells. More layer of granulosa cells. More layers of granulosa cells. Right? And one more thing here, very important, that here outside granulosa cells there is a theca layer. You can see yellow color. Theca layer will also form. Theca layer. Theca layer. Okay, theca layer. So this is very important. The secondary follicle. Now we are calling it secondary follicle. Primary oocyte, which is surrounded by more layer of granulosa cell, and one more layer present outside that theca layer, right? When primary oocyte, more granulosa layers, and theca cell, we can say it is a secondary follicle. Now it is called secondary follicle. This remaining you can forget, just it is a secondary follicle. Secondary follicle. Okay, secondary follicle, very very important. Now soon after this secondary follicle transform into tertiary follicle. You can see the secondary follicle, soon after it will transform into secondary tertiary follicle. So you can forget about this diagram I am taking here. This diagram I am taking here. So a little bit is it is very clear compared to that one. You can see on this, right? So now you can see this is the primary oocyte. Primary oocyte here also surrounded by more layer of granulosa cells. Okay, more layer of granulosa cells. Very very important, more layer of granulosa cells are there. Now here among the granulosa cells, there is a fluid filled cavity, right? So among this granulosa cell, you can see there is a fluid or liquid fluid filled cavity is there. I am showing with red color dots. This is the fluid filled cavity. Fluid filled cavity. So the fluid filled cavity we are calling antrum. Antrum cavity. Okay. Soon after, within the secondary follicle, among the granulosa cell, small cavity will form, right? So that cavity is filled by a liquid or fluid. So that is antrum cavity, right? Now these follicle, the theca layer differentiated into inner theca, that is called theca interna, outer theca, that is called theca externa. This is theca interna, inside, outside theca externa. Theca externa, theca interna. This is outermost layer, theca externa, and this innermost layer, theca interna. Question important. So how can you identify tertiary follicle? This is the tertiary follicle. Tertiary follicle. So this is tertiary follicle. Tertiary follicle. So how we will identify the tertiary follicle by identifying antrum cavity, and there will be two layers. Outer is theca externa layer and theca internal layer. Based on these characters, we can identify this is a tertiary follicle, right? So now this is very very important. Here primary follicle, secondary follicle, tertiary follicle. Within the primary follicle, within the secondary follicle, and within the tertiary follicle, there is only primary oocyte. Primary oocyte present. Here also primary oocyte. Here also primary oocyte. Very very important. Primary oocyte. Question. So from here they may ask you question. Okay. What is the common between primary, secondary, tertiary follicle? So these three follicles enclosing primary oocyte, which is arrested in prophase 1 meiotic division 1. But what is the difference? Here it is surrounded by only single layer of granulosa cells, but here it is surrounded by more layer of granulosa cell and theca layer. But here it is surrounded by more layer of granulosa cell, okay, but there is a cavity present in granulosa cell. Among the granulosa cell, there will be a cavity, and the theca layer is differentiated outer theca externa, inner theca interna. So these are the main differences between these three follicles. Okay, primary, secondary, and tertiary. Now, within the tertiary follicle, primary oocyte undergoes meiotic division 1. The meiotic division 1 will complete in primary oocyte, tertiary follicle. When primary oocyte undergoing meiotic division, when it was arrested in prophase 1, now it will start, right? So the primary oocyte, it continues the meiotic division 1 which was arrested in prophase, right? And it will produce a tiny polar body. It will produce a tiny polar body. I will try here. It will produce a tiny polar body and secondary oocyte. Okay, so primary oocyte, this is the primary oocyte completing meiotic division 1, producing a tiny polar body and secondary oocyte. Now you can say secondary oocyte is large in size because it is retaining bulk of the cytoplasm. The bulk of the cytoplasm from the primary oocyte is retained by the secondary oocyte. Okay, so it is an unequal division. Right? So both the gametes are not same, both the gametes are different. So spermatogenesis, we discussed that because of meiotic division we are getting two applied secondary spermatocyte, right? Because of the meiotic division 2 we are getting 4 applied spermatozoa or sperm. But in case of the oogenesis, when primary oocyte undergoing meiotic division 1, it can produce unequal division, one tiny polar body and secondary oocyte, one secondary oocyte. Okay, now soon after this tertiary follicle transform into Graafian follicles. Okay, so Graafian follicles or mature follicles. So this Graafian follicle ruptures and releasing secondary oocyte during the ovulation time. Okay, ovulation time. I hope it is cleared. Very important, oogenesis. Here the most of the important point you should remember, that is couple of millions of gamete mother cells are present in each fetal ovary. Their number never be added or formed after birth. You should remember characteristic features of the primary follicle, secondary follicles, tertiary follicle, and what kind of divisions are meiotic division 1 in case of the oogenesis. Unequal division, right? So we are getting one large body and small body. Large body we can say it is a secondary oocyte, one smaller one tiny polar body. Okay, so later this polar body will be degenerated and secondary polar body retaining bulk of the cytoplasm that helps in nourishment in future to the developing zygote. Okay, right. Here you can see one question. Statement 1. During the oogenesis, the first meiotic division occur in secondary oocyte. Is this right? The first meiotic division occur in secondary oocyte? No. The first meiotic division takes place in primary oocyte. Right? In primary oocyte, the first meiotic division takes place. So, the statement 1 is wrong or statement 1 is incorrect. Second statement, diploid secondary oocyte give rise to applied secondary pholioid body. No. Here primary oocyte 2n condition, total number of chromosomes are 46. Right? Oogonia also 2n condition, total number of chromosomes are 46. But here, okay, but here you can see meiotic division 1, meiotic division 1 or meiotic 1 is a reductional division. Reductional division, because of this reductional division, the chromosome number will be reduced to the original set. Okay, so then the primary polar body will get 23 chromosome and secondary polar body, secondary oocyte also get 23 chromosome. These are applied. So applied cells. So they are having only half of the chromosome to their original set, half of the chromosome. Okay, half of the chromosome. Right? So now based on this, now you can tell me diploid secondary oocyte. Generally secondary oocyte, just I told you, it is not a diploid condition. It is an applied condition, N condition. It gives rise to applied second polar body? No. Right? So generally second oocyte, secondary oocyte can produce applied second polar body, okay. But here secondary oocyte is not a diploid condition. Secondary oocyte is applied condition, but the secondary oocyte can produce applied secondary polar body. It can produce secondary polar body, okay, but the chromosome number is not 2n condition, but only n condition. So statement is both are incorrect statements. Both are incorrect statements. I hope you understood this one. Okay, so these kind of question they may ask you. So very important. So you can read NCERT line to line to get the concept. Very important. Right? So before going to this menstrual cycle, now you can see the secondary. This is secondary oocyte. Secondary oocyte. Now this secondary oocyte, it contains cytoplasm. Cytoplasm. Secondary oocyte contains cytoplasm and also nucleus. Nucleus present. Okay, nucleus. And within the cytoplasm, there are peripheral side of the cytoplasm, granules are present. These granules we are calling cortical granules. Cortical granules. Okay, cortical granules are present in the cytoplasm of the secondary oocyte. Generally, we can say it is also ooplasm. Ooplasm. We can also say ooplasm. Now, these secondary oocyte secrete a layer around it. Secondary oocyte secreting a layer around it. So this layer, you can see this is the layer secreted by secondary oocyte itself around it. So this layer we are calling zona pellucida. This is called zona pellucida. Zona pellucida. Right? Zona pellucida is a non-cellular layer, very important. Non-cellular. because it is not made up of with the cells, but it is made up of with the glycoprotein molecule. It is made up of with glycoprotein molecule. Glycoprotein molecules. So that's why it is a non-cellular layer. This layer is secreted by secondary oocyte itself. Okay. Now there is a gap or space between, and this is the plasma membrane of the egg. This is the plasma membrane of the egg. The space between plasma membrane and zona pellucida. The space between plasma membrane and zona pellucida we are calling perivitellin. Perivitellin membrane. Sorry, space or gap. Perivitellin space or gap. Right? In this perivitellin space or gap, first polar body present. The first polar body that came from the primary oocyte, when primary oocyte undergoing meiotic division 1, it can give rise to secondary oocyte, first polar body, and that first polar body present in perivitellin space. It is not given NCERT, but sometime they will ask you, because in the diagram NCERT it is mentioned that there is a cavity present between these two layers, plasma membrane and zona pellucida. Right. Next one, the innermost granulosa cells. These are innermost granulosa cells closely attaching to the zona pellucida. These are all innermost granulosa cells. Those are closely attaching to the zona pellucida layer. We are calling them corona radiata. Okay, so these are corona radiata. Corona radiata layer. So here from this diagram, you will get one question. They may ask you like this: arrange the following layer around the secondary oocyte. This is the secondary oocyte. First one, secondary oocyte surrounded by plasma membrane. Right? Plasma membrane. After plasma membrane, you can see there is a gap. Perivitellin space. So after this, there is a layer that is zona pellucida. This zona pellucida surrounded by corona radiata. Corona radiata. So this is the sequence: outer to inner, inner to outer. The sequence of the layer which are surrounded by ovum or secondary oocyte. Okay, so I hope you understood this. Very important. Mostly you remember this is the glycoprotein. It is a non-cellular layer, but corona radiata is a cellular layer. You can see these are all cells. Corona radiata is a cellular layer that is made up of with the granulosa cells. Right? Next one. So menstrual cycle, very important. So most of the question during since 2013 onwards, almost 10 or 15 questions are given up to 2024 NEET exam. The total number of questions from this topic related to human reproduction. This is the most important topic. Okay. So now here, menstrual cycle, right? So generally, reproductive cycle. The reproductive cycle which present in primates, female primates, we can say menstrual cycle, reproductive cycle. The reproductive cycle, so which present in female primates. Okay, The reproductive cycle which present in female primates we are calling menstrual cycle. Menstrual cycle. Okay. So example, menstrual cycle, example, apes, monkeys, and female human beings. Okay, female human beings. So in these primates, menstrual cycle takes place. The reproductive cycle or the cyclical event which takes place in the female reproductive system in female primates we are calling menstrual cycle, right? But in it is not there NCRT, but remember non-primates, non-primate mammals like dog, cat, rat, etc. These are the non-primates. In these animal, there is also one reproductive cycle present that is called estrus cycle. Estrus cycle. It is repeated every year or season according to the season. Okay. So generally during the estrus cycle, the concentration of estrogen hormone will be more in the blood. Estrogen hormone concentration will be more during the particular season. Because of that only sexual urge or sexual drive or they can menstruate, not it is a menstruation, so that is called generally EAT. The period is called EAT condition. Okay. Now menstrual cycle. Menstrual cycle, it typically it is 4 stages are present in menstrual cycle. Okay. The first one, menstruation phase. Menstruation phase, right? Second one, follicular phase. Follicular phase. Third one, ovulation. Ovulation. And the fourth one, luteal phase. Luteal phase. So these are the 4 stages that present in menstrual cycle. So but here very important, the other names of these phases also you should know, that is very important. Menstruation phase also called menses. Menses are bleeding phase. Bleeding phase. And follicular phase also called proliferative 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 phase, also called pre-ovulatory phase. Pre-ovulatory phase, very important. Pre-ovulatory phase, proliferative phase, and follicular phase, all are equal. And ovulation, okay, only ovulation is there. Next one, luteal phase, also called secretory phase. Secretory phase and post-ovulated phase. Post-ovulation, the phase of post-ovulation phase. Another name also very important. Sometimes they will ask you, follicular phase is followed by, or ovulatory phase followed by, right? Post-ovulatory phase or secretory phase, luteal phase, they will ask you. So other names also very important, you should know, right? Right. So this diagram of NCRT very important, and so keep concentration and very important, right? So first one, menstruation phase, right? It present 1 to 5 days. The 1 to 5 days is called menstruation phase. During this period, the endometrium lining is coming out in the form of menses or menstruation. During this phase, the highly developed endometrium lining is coming up in the form of bleeding or menses. Reason, right? During this menstruation phase, you can see the concentration of the estrogen hormone, concentration of the progesterone hormone. The concentration of estrogen hormone, concentration of progesterone hormones are very less, right? Because of the low concentration of estrogen, low concentration of progesterone hormone, the endometrium lining will not be maintained. The endometrium lining will not be maintained. So the endometrium, which is highly developed, along with uterine tissue and their blood vessel, they are coming out in the form of menses. Okay, so that is menstruation, right? The menstruation occur due to the released ovum is not fertilized. Suppose this is the ova which is released during the 14th day of the menstrual cycle. If this ovum is not fertilized, if the ovum is not fertilized after the ovulation, right, and this menstruation takes place. Lack of menstruation means that maybe pregnancy or stress, poor health condition or poor health condition. So these are the some reasons for the menstruation or menses. So question sometime they will ask you simple, which of the following condition is not present, or which of the following condition present during the menstruation, right? So what happening during the menstruation endometrium lining is coming out, right? And lack of menstruation, why menstruation taking place, that may be pregnancy or lack of menstruation means pregnancy. If the menstruation is there, that means ova secondary oocyte is not fertilized. Some other reasons also there, stress or poor condition. Lack of menstruation means these condition also affecting the menstruation. When this menses takes place or menstruation takes place, concentration of estrogen, concentration of progesterone hormones are very less. That time endometrium will fall out. Okay, or when the ovum is not fertilized, the released ovum is not fertilized, then only menstruation takes place. Right? Now, there are two cycles are present. Right? One, uterine cycle, that means the changes taking place in the endometrium lining during the menstruation. That is called uterine cycle. You can say this is uterine cycle. Right? The changes which are taking place in endometrium lining, we are calling uterine cycle. Next one, you can see this is ovarian cycle. The changes which takes place in ovary, right? So we are calling it ovarian cycle. There are two cycles simultaneously simultaneously takes place during the menstruation. One, the changes in the uterus or uterine endometrium, we are calling uterine cycle, and the changes in the ovary we are calling ovarian cycle. The changes in the uterine cycle, the changes in the ovary controlled by pituitary hormones, ovarian hormones. The hormones released by the ovary and the hormones released by the pituitary gland. So these are the responsible for the changes that takes place in endometrium lining and also that changes takes place in ovary. Very, very important. Okay. So now, so next one, 1 to 5 days menstruation takes place. So the first menstruation that takes place at the age of the puberty or 12 years. At the age of the puberty or 12 years, we are calling menarche. menarche. The first menstruation which takes place at the age of the puberty we are calling menarche, right? And menopause. Menopause is a condition where there is no menstruation. Menstruation will stop around the age of 50 years. That is called menopause. These two words are related to menstruation. Menstruation, the first stage of the menstrual cycle. Right. Now you can see. So there is a decreased concentration of progesterone and decreased concentration of estrogen in the ovary. When the concentration of progesterone, concentration of estrogen decreased, the low concentration of these two hormones, estrogen and progesterone, sending signal to anterior pituitary gland and also hypothalamus. Hypothalamus. The low concentration of progesterone hormone, low concentration of estrogen hormone in the circulation, sending sending signal, positive signal, or sending the signal to hypothalamus and anterior lobe of the pituitary gland. Then hypothalamus releases gonadotropin releasing hormone. Hypothalamus releasing gonadotropin releasing hormone. Gonadotropin releasing hormone stimulates anterior pituitary gland, right? So anterior pituitary gland releasing gonadotropins, follicle stimulating hormone, LH hormone. Okay, so follicle stimulating hormone, LH hormone. We are talking about follicular phase. Okay, so immediately after menstruation, the low level of progesterone, low level of estrogen stimulating or sending signal to the hypothalamus, anterior pituitary gland. So now hypothalamus releasing GnRH hormone and also stimulating anterior pituitary gland. GnRH stimulating anterior pituitary releasing two gonadotropins. Gonadotropins. Gonadotropins, that is follicle stimulating hormone, luteinizing hormone. Right? Now that follicle stimulating hormone working on primary follicle. Already I told you, primary follicle which is arrested in prophase 1 at the time of the, right? So primary follicle. Now these are the primary follicle, right? So each ovary contains 60,000 to 80,000 primary follicles. So right. So I didn't tell there. So because here only I am explaining that each ovary at the time of the puberty contains 60,000 to 80,000 primary follicles. Primary follicle. You know that primary follicles contain primary oocyte. That primary oocyte is arrested in prophase 1. Right? Now, because of the concentration of follicle stimulating hormone. Now this follicle stimulating hormone stimulating the primary follicle. Slowly primary follicle developing into secondary follicle. You know secondary follicle, more granulosa layer. So when this follicle is developing, this developing follicle releasing small amount of estrogen hormone. Very small amount of estrogen hormone. You can see slowly the concentration of estrogen hormone increasing. Concentration of estrogen hormone slowly increasing. Right? So the concentration of estrogen hormone slowly increasing towards the follicular phase. One more time. Follicular stimulating hormone stimulating the development of the primary follicle into secondary follicle. When this primary follicle developing into Graafian follicle, it releases some amount of estrogen hormone from the theca cells or granulosa cell. Now you can see concentration of the concentration of estrogen hormone increasing slowly towards the follicular phase. When the concentration of estrogen hormone increasing, you can see the concentration of follicular stimulating hormone LH hormone attaining peak level mid of the cycle, that is at 14th day. Mid of the cycle at 14th day. Okay, attaining maximum mid of the cycle at 14th day. You can see this one. Now here at the 13th day or 12th day of the follicular phase, concentration of estrogen hormone is more. The concentration of estrogen hormone is more at 12th day or 14th day of follicular phase. The high concentration of estrogen, sending signal to anterior pituitary gland to release LH. Okay, and also it is sending signal to the Graafian follicle. So Graafian follicle became matures. Maturation of Graafian follicle controlled by the estrogen. So now high concentration of estrogen stimulating maturation of the Graafian follicle and also stimulating anterior pituitary gland. So now the anterior pituitary gland releasing high amount of the LH hormone, right? So now at 14th day of the menstrual cycle or mid cycle, 14th day, mid of the menstrual cycle, the concentration of LH hormone will be more, high concentration or peak levels. So increase in LH hormone concentration during the 14th day of menstrual cycle, we are calling LH surge. LH surge. Okay, because of this high concentration of LH, is working on mature Graafian follicle. Now this mature Graafian follicle undergoing rupture, and that ruptured follicle releasing secondary oocyte, that is ovum, right? So the release of the secondary oocyte or ovum from the Graafian follicle with the help of the LH hormone, we are calling ovulation. That takes place 14th day, right? Right. This is about follicular phase. So now you can see what happens during the follicular phase. I will tell you. During the follicular phase, right, slowly in ovary, the changes that taking place in ovary are primary follicle developing into Graafian follicle, one change, right? Concentration of estrogen hormone increasing towards the follicular phase. Okay, concentration of follicular stimulating hormone LH hormone, these together called gonadotropins. So gonadotropin hormones are also increasing towards the follicular phase. Now when concentration of estrogen hormone is there, that estrogen hormone stimulating endometrium lining, right? Slowly endometrium lining is also proliferating, generating. So endometrium lining also proliferating or generating. Okay, for the development of the endometrium lining we need estrogen. That estrogen is secreted by growing follicles. Okay, the development of the growing follicles are controlled by gonadotropins. The release of the gonadotropin controlled by low level of progesterone, low level of estrogen. Okay, so these are the changes in uterine cycle. Endometrium lining is regenerated through the proliferation. Proliferation means rapid increasing. Okay, in ovarian or in ovary or ovarian cycle, primary oocyte or primary follicle developing into Graafian follicle, right? And gonadotropins are increasing towards follicular phase. But mid of the cycle, high concentration of LH because of high concentration of estrogen. So that is called LH surge. So mainly LH is working on mature Graafian follicle and stimulating them to undergo rupture. So they release secondary oocyte, right? So the secondary oocyte. After release of the secondary oocyte, the remaining part of the Graafian follicle converted into corpus luteum. Corpus luteum. Once that corpus luteum is acting as an endocrine tissue, temporary endocrine tissue. Now these corpus luteum will produce progesterone hormone. You can see. So concentration of progesterone hormone is increasing. When concentration of progesterone hormone is there in the blood, that stimulate endometrium lining, thickening of the endometrium lining. So then endometrium lining will be thickening and endometrium lining will be maintained for implantation. So that's why progesterone is called pregnancy hormone. Now, this is luteal phase. What happening during the luteal phase? Corpus luteum, that Graafian follicle, the remaining part of the Graafian follicle converted into corpus luteum, right? Endometrium lining became thick, right? And because of concentration of progesterone hormone is more. And these LH also maintain corpus luteum. High concentration of LH maintains the corpus luteum. When LH is maintained the corpus luteum is secreted by the corpus luteum secreting progesterone. So that progesterone is working on endometrium lining and endometrium lining is maintained. Okay. So later when concentration of endometrium is high, the high concentration of endometrium sending negative signals to the LH hormone, right? In the high concentration of progesterone hormone sending negative signal to the anterior pituitary lobe to not to release LH. When LH is not releasing, endometrium is not maintained. So it is converted into corpus albicans. Okay. Now there is no release of the progesterone. So again progesterone levels are decreasing. So when progesterone levels are decreasing, this endometrium line will not be maintained. It will come out in the form of menses. Okay, these are the changes that takes place during the menstruation. Right? So now here you remember luteal phase is a fixed 14 days. No change. It is fixed days in luteal phase. It remains 14 days for 14 days. Ovulation takes place at 14th day. So generally proliferative phase is a variable phase. Proliferative period or proliferative phase is a variable phase because of it varies person to person. Right? Generally it is a 8 days or 8 to 9 days. And you know menstruation period is 1 to 5 days, but it lasts 3 to 5 days. The bleeding or menstruation takes place 3 to 5 days. Okay. Now why we are calling this follicular phase? Because of there is a development of follicle in ovary. Why we are calling it proliferative phase? During this phase only endometrium lining is proliferating and developing. So that's why it is called follicular phase, proliferative phase. This is the phase which is taking before to the ovulation. So we are calling pre-ovulatory phase. So ovulation means release of ova. Next one. This is called luteal phase because corpus luteum present. And secretory phase because it secretes endometrium lining secretes some secretion. Okay. So that's why it is called secretory phase. And this corpus luteum also secrete very small amount of the estrogen. You can see very small amount of estrogen also released by the corpus luteum and also relaxin. Small amount of the corpus estrogen and relaxin are the two hormones released by the corpus luteum. Okay, any doubts? I hope it is understood this concept very. Okay, 1 minute. Okay, I hope this menstrual cycle concept is clear for everyone. and you can see the one question, 2024 question. I think it is in NEET exam. NEET assertion. You can do this. Follicular stimulating hormone act upon ovarian follicle in females, right? Leydig cells in male. Is it right? Okay, just I told you that follicular stimulating hormone is acting on ovarian follicle. You can say it is right. Is it working on Leydig cells? No. Why? Leydig cells are controlled by LH hormone, are Leydig cells, right? LH hormone that stimulate interstitial cells, Leydig cells that present in intertubular space in the inside the testis, that is in between seminiferous tubule. These Leydig cells secrete testosterone hormone. Okay, testosterone hormone. But follicle stimulating is hormone. In case of the male, it can stimulate Sertoli cells, not Leydig cell. Okay, so follicle stimulating hormone in case of the male, it do not stimulate Leydig cell, but it can stimulate Sertoli cell or sustencular cell. They can secrete androgen binding protein and inhibin protein, right? So now assertion is wrong. Reason: growing ovarian follicle secrete estrogen. Just I told you in female, right? While interstitial cell secrete androgen in male human being. Just I told you. Interstitial cells are also called Leydig cells. Interstitial cells are also called Leydig cells. So Leydig cells secrete male androgen testosterone, right? So ovarian follicle also secrete estrogen. So statement reason is right, but assertion is wrong, right? So simply both are right, no. A and R right, no. A is true, no. R is false, no. A is false, yes. And R is right, yes. Okay, so this is the question that is given in 2024 NEET exam, I think. I hope you understood this question. Next one more question related to the menstrual cycle. Which of the following events not associated with ovulation in female, right? So during the ovulation, just we had discussed that LH surge, high concentration of LH stimulate mature Graafian follicles to release ovum. So this is right. They are asking not associated. This is associated. And full development of Graafian follicle, yes, Graafian follicle. Tertiary follicle will develop into Graafian follicle, mature follicle. This is also right. Release of secondary oocyte, yes, after rupturing, secondary oocyte releasing, Graafian follicle releasing secondary oocyte, right? Now decrease in estradiol, no, this is wrong. Increases in estrogen concentration, you can see in the diagram. You can see during the ovulation time, concentration of estrogen hormone is more, right? So another name for estradiol, estrogen, estradiol. So that's why not decreases, it increases. During the ovulation time, mid of the cycle, concentration of estrogen is more. When estrogen concentration is more, it is sending signal to the anterior lobe, that is releasing LH hormone. So LH is working on Graafian follicle. Graafian follicle is releasing secondary oocyte from the matured follicle. Okay, so this is the wrong one and this is the correct answer. Not associated. Okay, next one. No new follicles follicle develop in luteal phase of menstrual cycle because. So this is the NEET 19 question, corona period time. Both which of the following event? No new follicle develop in the luteal phase. Yes. Why? Both LH follicle stimulating levels are low in the luteal phase. Just you can see the diagram and tell. And follicle do not remain in the ovary after ovulation. No, just I told you 60 to 80,000 follicles will remain. Okay. So this is simply it is wrong. LH levels are high during the luteal phase. Bro. No. You can see this is the luteal phase. Progesterone levels are high. Very small amount of estrogen present. Right? So LH levels and you can see LH is also more. And you can see here follicle stimulating hormone also more. So this is the right answer. Because of high concentration of LH hormone, high concentration of follicle stimulating hormone in luteal phase, right? That do not allow the primary follicles to develop into Graafian follicle. The high concentration of LH, sorry, low concentration of LH and low concentration of follicle stimulating hormone. Because these are the hormones that can help in development of the follicles. If the concentration of these hormones are very less, how could they help in development of the follicle? Okay, so during the luteal phase, concentration of these hormones are very less. Later phase, not initial period of the luteal phase, later phase of the luteal phase. So answer is A. Right? Next, fertilization and implantation. Fertilization and implantation. Okay, so male inseminate during the coitus or copulation time. Copulation time or during the coitus. Male partner inseminate semen in vaginal cavity or female reproductive tract. Vaginal cavity or female reproductive tract, you can see. Okay. Now, male partner ejecting semen in female vaginal cavity. Now this is through the cervix and uterus, it is moving to the ampullary region. So now you can see they are moving to the ampullary region. These are the sperms they are reaching. This is the ampullary region. So ampullary region, you can see secondary oocyte released from the ovary also moving towards ampullary region. Right? So 14th day ovulation will be there. After that, the released ova collected by the fimbriae and ova also really moving towards ampullary region, and the sperm also moving towards ampullary region. The route you remember: vaginal cavity, cervix, uterus, isthmus, and ampullary region. So this is the region where fertilization takes place, right? Where fertilization takes place. But all copulation do not lead to the fertilization. The copulation in which the movement of the sperm and movement of the ova simultaneously should go to the ampullary region. Okay, we require simultaneous movement of the sperm and ova towards ampullary region. These kind of movements only that lead to fertilization. fertilization. Question important. All fertilization, sorry, all copulations, all copulations do not lead to the fertilization. The copulation where or in which there is a simultaneous movement of the egg and the sperm toward the ampullary region, that type of copulations are lead to fertilization. Okay, so after fertilization, the zygote is formed in ampulla region. Very, very important. Ampulla region. You can see this is fertilization, right? So fertilization taking place in ampulla region. Already I explained about this diagram. This is ovum, plasma membrane, and corona radiata, and this is zona pellucida. Green color perivital space. Zona pellucida, cells of corona radiata. Now, during the fertilization time, right, if the sperm touching with plasma membrane, sorry, zona pellucida, when the sperm touching the plasma membrane, these plasma membrane undergoing some changes when the sperm touching the zona pellucida layer of the secondary oocyte, right? So then the zona pellucida layer undergoing some changes. The changed zona pellucida layer does not allow any extra sperm into the inside of the secondary oocyte. Okay, now you can, this is very, very important. That's why it is prevented. Polyspermy is prevented. Entry of the more sperm into the ovum is prevented because it undergoing some changes. So we are not going into the deep that anti-fertilin, fertilin, okay, calcium ions and cortical granules. So we are not going into the deep. Just we are NCERT line. I will teach you that. Only NCERT line. You can see. Just you remember when the sperm touching the zona pellucida undergoing some changes. The changed zona pellucida does not allow any extra sperm into the ovum. So that's why it is preventing polyspermy. Right? Later, the acrosome of the sperm, releases certain enzymes. Right? That enzyme helps the sperm to enter into the cytoplasm. This is the cytoplasm. Okay, enter into the cytoplasm via penetrating through the corona radiata and plasma membrane. It making the way via zona pellucida and plasma membrane and enter into cytoplasm. When it enter into the cytoplasm, right? So secondary oocyte, you know that it is arrested in metaphase 2. Primary oocyte, which is arrested in prophase 1, completing meiotic division and producing second polar body. First polar body and secondary oocyte. This secondary oocyte is also undergoing meiotic division 2, but it is arrested in metaphase 2. Now that metaphase division 2 will complete when the sperm enter into cytoplasm. When the sperm enters into the cytoplasm of the secondary oocyte, now the secondary oocyte completing that meiotic division 2 and produce ovum or otid and second polar body. Right, chromosome number here also 23. Reason meiotic division 2 is equal to mitotic division. There is no reduction in the chromosome number. It is like a mitosis. So chromosome number will be 23. Now you can see this is the ovum and this is the nucleus of the ovum and this is the sperm head applied nucleus. This is the nucleus of the sperm. Now the nucleus of the sperm and the nucleus of egg undergoing fusion, that is called fertilization. The fusion of the male and female applied nucleus, right? So they produce zygote, diploid zygote. That is fertilization takes place in ampulla region, right? Now After fertilization, you can see this is the fertilization, ampulla region. After fertilization, now the zygote is formed. When the zygote moving through the ampulla isthmic junction or isthmic region, you can see this is isthmus. When the zygote moving through the isthmus of the fallopian tube, zygote undergoing division, you can see zygote undergoing mitotic division. So the mitotic division that takes place in the zygote while it is moving through the isthmus, we are calling them cleavages. Question important. So cleavage takes place in zygote, and when that zygote moving through the isthmus, right? So it can produce 2-cell stage, 4-cell, 8-cell, 16-cell stage of the zygote, right? Right? Now, when the zygote 8 to 16 cell stage, 8 to 16 cell stage, it looks like a mulberry fruit. You can see here. Zygote, when it present in 8 to 16 cell stage, it looks like a mulberry fruit. So we are calling it marula stage. Mulberry fruit, so we are calling marula. Now these marula from the isthmus, further it enter into uterus. Further it enter into uterus. When it enter into uterus, the blastomers means simple the cells. Because of the division, the zygote is inside the zygote, smaller and smaller cellular units are forming. That smaller and smaller cellular units we are calling blastomers, right? When the zygote 8 to 16 cell stage blastomers we are calling marula. When these marula from isthmus enter into uterus, right? So these blastomers are arranged into two layers. So these blastomers are arranged into two layers. The outermost layer we are calling tropoblast layer. Outermost layer is tropoblast layer, and some cells of the blastomers attaching to the tropoblast layer we are calling inner cell mass. Inner cell mass. Okay. Now this tropoblast layer helps in attachment of this embryo with uterine wall, that is called implantation. Attachment of the embryo with uterine wall we are calling implantation. But these inner cell mass differentiate into embryo. Inner cell mass differentiate into embryo, and inner cell mass can develop into ectoderm, mesoderm, endoderm. Okay. So inner cell mass will differentiate into ectoderm layer, endoderm layer, and mesoderm layer, and these layer produces all types of tissues, organs, tissues and organs. Okay. These inner cell mass contain some stem cells. They has capacity of totipotency. Stem cells also present. Stem cells totipotency. Totipotency cell, they has capacity to develop into complete organism. So we are calling it totipotency cell. Okay. So this is development of the embryo zygote. Next one. Pregnancy. Pregnancy. So this is endometrium lining. Endometrium lining. Now this is outermost layer, tropoblast layer, and this is inner cell mass. Inner cell mass. After implantation. Implantation means attachment of the embryo in the blastocyst stage to the uterine wall is called implantation. Okay. So now you can see this tropoblast layer forming small finger-like projection we are calling chorionic villi. Chorionic villi. chorionic villi. The trophoblast cells are forming chorionic villi. Now, maternal, this is endometrium lining and the blood vessels of the blood vessels and maternal tissue or uterine tissue. Chorionic villi, uterine tissue and its blood vessel together forming a structural functional unit between developing baby and mother, that is called placenta, right? Placenta. So now later it is developing like a structure. This is placenta between developing fetus and maternal body, mother. So this structure we are calling placenta. This placenta connected to the baby by this structure called umbilical cord. This is umbilical cord. Umbilical cord. Umbilical cord is a structure which is connecting the placenta with mother and fetus, right? So these placenta releases hormones. Very, very important. The hormones released by the placenta. 1. HCG, HPL, relaxin. Okay. So human choriogonadotrophin hormone, human placental lactogen hormone, relaxin hormone. Okay. So these are the hormones released by the placenta, but one exception here you can see, relaxin is an hormone released by the ovary. I told you corpus luteum, released by the ovary during the later phase of the pregnancy or at the end of the pregnancy, at the 9-month period of the pregnancy. Relaxin. These are the hormones only present in the blood during the pregnancy. Only hormones that present only during the pregnancy, they appear in the circulation. Only these hormones. But certain hormones concentration will increase during the pregnancy. Those hormones are estrogen, okay, progesterone, cortisol, thyroxine, prolactin. Normal condition, during the normal type, the concentration of these hormones are present but less. But during the pregnancy, the concentration of these hormones will increase in the blood several folds. Okay, the concentration of these hormones will increase more during the pregnancy time, right? And these hormones, they support for the growth of the baby or fetus, fetal growth, and the changes, the metabolic changes that takes place in mother during the pregnancy, also supported by the these hormones. So these hormones also support the metabolic changes occur in mother, right? And also they maintain the pregnancy. These hormones helps in maintaining the pregnancy, maintains pregnancy. Question very, very important from here. They may ask you, right? And I think 2019 or last year one question they asked that later phase of the pregnancy, which of the following hormone is released? Instead of giving ovary, they have given that corpus luteum. Okay, because corpus luteum is the part of the ovary. So the right answer is corpus luteum. It is given question in 2020 or 21. Okay, so these are the hormones their concentration will be increasing more during the pregnancy. Why? Because they has to support for the fetal growth, and metabolic changes occur in the mother are supported by these hormones, and also they maintain pregnancy. Okay, next one, organogenesis or gestation period, major events during the gestation period in humans. Organogenesis. Okay, so human pregnancy or gestation period is 9 months. It is divided into 3 trimester. Trimester 1, trimester 2, 3. The first trimester, okay, it is 12 weeks. The first trimester of the gestation period, or human gestation period, 9 months divided into 3 periods, right? So first one, the 4 week of the pregnancy, or during the first trimester, 4 weeks. 4 week means 1 month. 1 month fetus, heart will form. Okay, so the sign of the growing fetus can be noticed by listening to the heart sound through the stethoscope. Okay, so the first month or 1 month of the fetus, heart is formed in 1 month of the fetus. 1 month you remember, 4 weeks also you remember, very very important. Okay, so first sound can be observed in growing fetus by listening through the stethoscope, very very important. Next one, right? End of the second month, end of second month or 8 week, end of second month, 8 week of the fetus develops limbs and digits. Okay, limbs and digits will develop, limbs and digits. Next one, end of the third month, end of the third month or 12 week, or first trimester, end of the first trimester, end of the 3 months, end of the 12 week of the pregnancy, most of the major organ systems like, right, external genitalia, limbs are formed. Most of the external genitalia and limbs are formed. This is very 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 important. Okay, first month, second month and third month. Next month, second trimester, okay, so from 12 weeks onwards, okay, 20, week number 20 and fifth month of the fetus, the first movement of the fetus and appearance of air on head. You can see the first movement of the fetus, appearance of airs on head will be observed during the fifth month of the fetus. Fifth month of the fetus, there will be first movement of the fetus and appearance of air on head. Okay, so end of the 24 week or second trimester, okay, second trimester, body is covered with fine hairs, eyelid, eyelashes are separated. So the body is covered by fine hairs, eyelid, eyelashes are separated. Very very important. Next one, so last third trimester, 36 weeks, 9 months. Okay, so 36 weeks or 9 months. Okay, fetus is fully developed and ready for delivery. The fetus will be fully developed, you can see here, fully developed fetus and ready for delivery. Right, ready for delivery. Next, parturition. The process of childbirth is induced by the neuroendocrine mechanism. Delivery of the baby is called parturition. It is a neuroendocrine mechanism. That means neural system as well as some hormones are involved in parturition, expulsion of the baby from the uterus. Right? So here right, this is the question we will discuss later. You now you can see. Okay, fully developed baby, the full developed fetus and placenta. This is full developed fetus and placenta. Full developed fetus and placenta sending signal for parturition. Sending signal to the, you can say this is full developed baby. The full developed baby sending signal for parturition or originate. When baby is fully developed, that is sending signal. So then the uterus undergoing mild contraction. Uterus undergoing mild contraction. When uterus undergoing mild contraction, that is called fetal ejection reflex. Fetal ejection reflex. So these fetal ejection reflex, you can see sending signal to the hypothalamus. So hypothalamus posterior part is releasing hormone from mother. That hormone stimulating uterus or working on uterus. So again uterus undergoing contraction. That contraction again sending signal to the hypothalamus. Hypothalamus again releasing oxytocin. Oxytocin again working on uterus muscle. So these repeated steps helps in expulsion of the baby through the birth canal. Through the birth canal, we are calling that. After delivery, the placenta also coming out from the uterus. Okay, so after delivery also placenta is coming out through the uterus. Right, next lactation. End of the pregnancy. Right, so during the end of the pregnancy, mammary glands are differentiated. Towards the end of the pregnancy, mammary glands are differentiated and they are ready for production of the milk. Now you can see the process of formation of milk at the end of the pregnancy, right? Bringing up the healthy baby. Example, right? Sucking up the baby, sending signal to the hypothalamus, right? Hypothalamus neurosecretory cells are releasing, right? One hormone called oxytocin. That oxytocin hormone is stored in posterior part of the pituitary gland. This is oxytocin, posterior part of pituitary gland releasing oxytocin, anterior part of the pituitary gland releasing prolactin. Prolactin hormone. Prolactin hormone working on mammary alveoli and alveolar cell stimulates synthesis of the milk. Synthesis of the milk. Oxytocin causes the contraction of the muscle that present around the alveoli, right? So prolactin helps in production of the milk. Oxytocin helps in ejection of the milk, right? So the milk which is secreted by the mother during the initial days of the lactation, that is called colostrum, right? So colostrum is rich in IgA antibody. You can see IgA antibody is a dimer. It is made up of with two antibody molecule, dimer molecule. Okay, so colostrum is rich in IgA antibody, right? So now you can see this is the question that is given from that area. Breastfeeding during this is given, I think 2020-24 last year NEET exam. During the initial Initial period of infant growth is recommended by the doctor. Breastfeeding, right? Bringing a healthy baby, right? Colostrum contains several antibodies, absolutely essential to develop resistance for the newborn. Here both are right, and R is the correct explanation of A. R is the correct explanation of A. So this is the question that is given in 2024 NEET exam, I think. Okay. Right. So next one, we completed that human reproduction chapter. Next we are taking that the small chapter and also very important. So human reproduction and health. Right. So this definition important according to WHO. What is reproductive health? So the total well-being in all aspects related to reproduction, that is physical, emotional, behavioral, social, in relation to reproduction is called reproductive health, right? So family planning program was initiated by Indian government in 1951. Very important. India is the only country that is initiated family planning program in the world, right? Now this family planning program, now it is changed into reproductive child health care RCH, because improved programs are covering wider reproduction related areas. Now these are currently working under RCH. This is important. The importance of the RCH program is it creates awareness among the people about various reproduction related aspects and providing facilities support for the healthy reproductive society. RCH main function create awareness about the reproductive related problems. Okay, that also you remember. Okay, next one. To create awareness among the people, what we have to do? Print media, paper, right? Society, school, governmental, non-governmental agency, friends, teachers, and family, right? Everyone should participate in creating awareness among the people. This is also important. And introduction of the sex education in school. By introducing the sex education in school, it provides proper information to the young ones about reproduction and reproductive related problems. And sex education helps the children to discourage the myth, right? Believing the myth and misconception about sex related aspects. And also it provides correct information about sexually transmitted diseases. Okay, and it also helps in proper information about reproductive organ. By introduction of the sex education, it tells about the proper information about reproductive organs, about adolescence and related changes, safe, hygienic sexual practice, and about sexually transmitted disease like AIDS, right? These would help the people, especially those are in adolescent age group, to create reproductively healthy society. Okay, so this is the dry chapter, line to line you have to read and you have to focus on these also. Next, people, especially those are in fertile couple and those are in marriageable age group. So now we have to educate the people those are in marriageable age and fertile couple about available birth control options and care of pregnant mother, postnatal care of mother and child, importance of the breastfeeding, equal opportunities for male and female child. Okay, so these kind of awareness program we should, right, create among the especially in fertile couple and marriageable age. Okay, so implementation of various action plans that help us to achieve reproductively healthy society, right? So strong infrastructure facilities should be provided. That means building of hospitals. Professional expertise, that means recruiting more and more expert doctors. And material support, we should supply all the material that is needed. Example, ECG, right? Birth options, uh, right? So material support. That next one, medical assistant, okay, and they should tell problems like pregnancy, delivery, STD, abortion, conception, menstrual problems, so infertility. These are the problems that will be discussed by the doctors, or by creating awareness by implementing the various action plans and government and non-governmental agency. Okay, so this is very very important. Amniocentesis. So amniocentesis is a technique where collecting amniotic fluid. So this is the growing fetus. Growing fetus surrounded by the layer called amnion. Inside that there is a liquid called amniotic fluid. So right, these amniotic fluid contains certain proteins, certain enzymes, right? So now we are collecting this amniotic fluid, right? And by examination of these amniotic fluid cells, we can identify in growing fetus in future they may suffer with Down syndrome or not, hemophilia, sickle cell anemia. Okay, so generally we are using this amniocentesis technique to identify the presence of certain genetic disorders like hemophilia, Down syndrome, sickle cell anemia. But these technique and also the survivability of the fetus, right? The survivability of the fetus also can be determined by using the amniocentesis. Very very important. Which of the following function is not related to the amniocentesis? They will ask you. Amniocentesis can be used to the detect. So these are the techniques. Now it also can reveal the sex of the baby. This is the main problem with amniocentesis. If we are using amniocentesis technique for the detection of the genetic disorder, that is okay. But most of this time, this technique is used to detect the sex of the baby. So that's why Indian government is banned amniocentesis technique for the purpose of sex determination. For the purpose of sex determination, this technique is banned. Reason, illegally, legally, we can check, and increasing female feticides, abortions. Okay, this is the area from reproductive health chapter. You may get question, right? So population explosion and birth control. Now you can see population in the 19 year 2 billion population around the world. This data is very very important, right? In India, so data is not given, but approximately 350 millions of population at the time of independence. So at the time of independence, 1947. In 2000, the world population 6 billions. In India, it is close to 1 billion, right? May 2011, it crossed 1 billion, crossed 1.2 billion, May 2011. Okay, 7.2 billions world population, but India 1.2 billion population. Okay, so earlier the growth was 1.7, now it is the growth of the Indian population less than 2%, less than 2%. The reason for the growth, explosion of the population reason, increased health facilities and decreased maternal mortality rate, decreased infant mortality rate, and health conditions. So these are the some factors. Because of those factors, the population is increased. Okay, right. So how to control the population? Contraceptive method. Contraceptive method. Contraceptive methods, they prevent conception. Physical or meeting of the sperm with egg is called conception. Contraceptives generally they prevent contraceptive they can prevent pregnancy by interfering with ovulation, ovulation, implantation, implantation, and changing the cervical mucus lining, cervical mucus lining. Okay, so how contraceptives are working? Contraceptives they can interfere with normal functioning of the ovulation, implantation, and change in the cervical mucus and fertilization. Fertilization, right? The contraceptive methods are natural or traditional method. Example: periodic abstinence, coitus interruptus, lactational amenorrhea. Surgical method: vasectomy, tubectomy. Injections and implants, barrier methods, and oral contraceptives, intrauterine devices. Okay, so briefly we will be discussing here, and very important also. This is very important. Okay, now periodic abstinence means, right? Periodic abstinence, it is a during 10 to 17th day of the menstrual cycle, where in this period or in between these days ovulation is expected. Ovulation is expected. So that's why the couple should avoid coitus or copulation during this period of the menstruation, because ovulation could be expected. Ovulation could be expected, so it is a highly fertile period. Fertile period. Because of that reason, the couple should abstain or avoid coitus during that period. That is called periodic abstinence. Coitus interruption means male partner withdraws his penis before ejaculation or before insemination into vaginal cavity. So that is also coitus interruption. Lactational amenorrhea, during the lactation after birth, right? So after birth lactation is there. During the intense lactation period, because of high concentration of prolactin hormone, high concentration of prolactin hormone, right? So in the high concentration of prolactin hormone, there is no LH hormone, there is no follicle stimulating hormone. If there is no LH, there is no follicle stimulating hormone, there is no development of the follicle, right? So this period the couple can use for coitus. Okay, and this is very effective only up to 6 months. Lactational amenorrhea, amenorrhea means absent, menstrual cycle will absent up to 6 months. So because of there is no development of follicle, high concentration of prolactin inhibiting release of the follicle stimulating hormone, LH hormone. Okay, these are natural methods. In the natural method, we are not using any devices or any chemicals. Any chemicals. The chance of side effects are nil, but chance of failure also very high. The risk of failure is also very high. That is very important. Which of the following contraceptive method, right, without any side effect or no side effect? That is natural method, traditional method, right? So there is no side effect because we are not using any drug or any device. So it is the safest method. But problem is high failure chance. Chance of failure also very high. Okay. Next, barrier method. Barriers, natural methods and barrier methods are both are working on the principle is avoiding physical meeting of the sperm with ovum. Okay, the main principle is avoiding of the physical meeting of the sperm with ovum, right? Barrier. We're creating a barrier between sperm and egg. There is no meeting of the sperm with egg. So we are preventing by using barriers. Example, condom, diaphragm, cervical valve. So these barriers are made up of with thin rubber, okay, and latex. So condom is made up of with thin rubber and latex, but diaphragm, cervical cap, valves are made up of with rubber only with thin rubber. Very very important. Okay. Now diaphragm, cervical cap, valve. These are the barriers generally used by the female. Generally used by the female to cover cervix. To cover cervix, and these are reusable barriers. They can be used again. Reusable barriers are diaphragm, cervical cap, valve. Right? These male condoms and female condoms are disposable. Male condom and female condom both are disposable, and they are made up of with thin rubber and latex, very important. But diaphragm, cervical cap, valves are made up of with only rubber. They can be used by the only female to cover the cervix. But these condoms are made up of with thin rubber and latex. They can be used by the male and female. Right? In female, it helps in covering of vagina and cervix, but in male it used to cover the penis. Male it is used to cover the penis, only the condom. The main example of the one male condom, Nirod is one of the familiar male condom. Female condom is called femidome. Femidome. So this is very 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 important. Okay. So nowadays there is an increase in the use of the condoms in case in Indians, due to condoms are not only protecting the not only helping in the contraception, but also they mainly helping in controlling or avoiding STD, transmission of STD, sexually transmitted diseases. So that's why the use of condoms is also benefiting to prevention of sexually transmitted disorder. Okay. Now the condoms also self insertable, so it can provide privacy. So that is one of the reason it use of condoms are increased in India. Okay, self insertable, they provide privacy. Okay, so these are the contraceptive methods. Next, intrauterine devices. Intrauterine devices, there is a small device that is inserted into the uterine cavity only in female. These intrauterine devices classified into non-medicated, copper releasing intrauterine devices, hormone releasing intrauterine devices. Lippis loop is example for the copper releasing intrauterine devices. You can see this is Lippis loop, right? So this Lippis loop inserted into female genital tract, uterus. So this Lippis loop generally it is made up of with the stainless steel or rubber. It is made up of with stainless steel or rubber. When they are inserting into the female genital tract, that is uterine cavity, so it is acting as a foreign body. It is acting as a foreign body, right? So it can attract phagocytic cells. It can attract phagocytic cells. So once the phagocytic cells are enter into uterine cavity, they can engulf sperms, right? So they prevent pregnancy. Copper releasing intrauterine devices. Example, copper T, right? Copper 7 and multi-load 316 375. These are very, very important. Copper releasing intrauterine device example, copper 7, copper T, multi-load 375. Main function, the releasing copper ions. When the copper ions are released by these, okay, these copper ions reacts with middle phase, mitochondria. Okay, copper ions are reacting with mitochondria, then mitochondria will be damaged in the middle phase. When mitochondria is damaged in the middle phase, it will not be provided, it will not provide any energy for the movement of the tail. So right, it decreases sperm motility. How it is decreasing sperm motility? The copper ions are reacting with mitochondria of the middle phase of the sperm. So when there is no mitochondria, there is no movement of the tail. If there is no movement, the sperm cannot move to the ampullary region. So they prevent fertilization. Next one. Hormone releasing intrauterine device. Example, progestastat, LNG20, levonar gestal 20. LNG means levonar gestal 20. Hormone releasing intrauterine devices. Okay. So these hormones makes these uterine cavities unsuitable. These hormones once released, they make cervix and uterine cavities unsuitable for the form, unsuitable for the movement of the sperm when these do not reach the ampullary region, and they prevent fertilization. Okay. Next one, oral contraceptive pills. So oral contraceptive pills, they are tablets. Oral contraceptive pills are tablets, right? Oral contraceptive pills are, they contain either progesterone, progesterone or estrogen, or progesterone estrogen combination. They are made up of with only progesterone, either progesterone estrogen combination. They are using in oral contraceptive pills or tablet. Now these oral contraceptive pills mainly affecting on ovulation, right, implantation, and alter the quality of the cervical mucus, inhibit the motility of the fallopian tube. Okay, when female employ, when females are employing this oral contraceptive pills, or when they take oral contraceptive pill, it can prevent ovulation. When concentration of progesterone hormone, estrogen hormones are more, right? Concentration of estrogen and progesterone more, that inhibit release of the follicle stimulating hormone, LH hormone. You know, already you studied in menstrual cycle. When concentration of progesterone is less, that's sending signal to the anterior lobe of the pituitary gland. Then they will release follicle stimulating hormone, LH hormone. But already in your blood, concentration of estrogen and progesterone is more, so they prevent release of these hormones. So there will be no ovulation. Implantation, because they alter cervical mucosa. Okay, cervical mucosa is altered. They prevent entry of the sperm. So the mucus is not suitable for the movement of the sperm in the uterine cavity, and inhibit the motility of the fallopian tube. When fallopian tube is not moving properly and there is no movement of the egg towards the ampulla region. Okay, right. Here, oral contraceptive pills are very effective, right? And most of the females are using with less side effects. Very, very important. Oral contraceptive pills, right? So very effective, effective with less side effects, less side effects, less side effects. Most of the females are using this. Most of the females are accepting this. Okay, and oral contraceptive pills prevent ovulation, implantation. Remember. Now, the combination of the progesterone or estrogen or only progesterone. Also, we are giving in the form of injection or implantation under the skin. The implants also working, the working of the implants also same. The injection contain progesterone, only progesterone or combination of estrogen and progesterone. The function of the injections and implant similar to the oral contraceptive pills. Oral contraceptive pills. But the injection and implants much more longer. Their effective is much more longer compared to oral contraceptive pill. So that is also important. So they can work up to 5 years. Much more long duration injection. Okay. Next, medical termination of pregnancy. So intentional or voluntary termination of pregnancy before its full term development is called medical termination of pregnancy, MTP, or induced abortion. Induced abortion. Okay. Now you can see induced abortion 45 to 50 million years MTPs are performed all over the India, that accounting 1/5 of total number of conceived pregnancies, right? So 40 to 50 millions of abortion MTPs are performed every year all over the world, that account the total 1/5 of the total conceived pregnancies, right? Now, so Indian government is legalized MTPs, right? And indiscriminate and illegal female feticide. Okay, na? And why or when can we suggest MTP? So avoiding unwanted pregnancy, to get rid of unwanted pregnancies due to casual intersexual intercourse, failure of contraceptive, continuation of pregnancy is problem for the mother and fetus during that time, during the rape or assault during the that time also, on advice of the doctor, two doctors, on advice of the two doctors, first time only one doctor, but on advice of the two doctor only we will suggest MTP, medical termination of the pregnancy, right? So only we will suggest MTP during the first time trimester or up to 12 weeks is the safest period. But if the second trimester also, if you wanted to terminate the pregnancy, you should consult or you should take permission of the two recognized medical doctors. Two recognized medical doctors. Okay. So the problems related with MTPs. Most of the MTPs are performed by the unqualified quick, and amniocentesis one of the reason to the MTPs. Because of the amniocentesis, MTPs are performed. Reason, amniocentesis is a technique generally we use it to detect the genetic disorder. Most of these amniocentesis illegally using to determine the sex of the baby. If frequently they are detecting the sex of the baby is female, they are performing FTP on female feticide, right? So now repeated MTP is also danger for the mother and fetus, right? So effective counseling should be needed, right? So this is about MTP. From these area also questions are expected, right? Next one, STDs, sexually transmitted diseases or sexually transmitted infections or venereal diseases. Sexually transmitted infections, STI, or venereal diseases, STDs. Okay, so there are bacterial, viral, protozoan disorders are there. So gonorrhea, Neisseria gonorrhea is a bacterial disease. Syphilis, Troponema pallidum also bacterial. Genital herpes, it is a viral, right? Simplex virus, human herpes simplex virus is responsible. Genital wart, cervical cancer caused by human papilloma virus. Trichomoniasis, this is a protozoan parasite, Trichomoniasis vaginalis, right? Protozoan parasite. Chlamydiasis, Chlamydia trachomatis, it is also bacterial. Hepatitis B, Hepatitis B virus, HIV, AIDS. Among these, cervical cancer, HIV, Hepatitis B, uncurable. But remaining all other diseases are curable if we detected early and treated properly. So they are curable. Okay. So the symptoms of the STDs, early symptoms, itching, fluid discharge, swelling in pelvic region. Okay. And these are some initial symptoms of the STDs. Most of the people, those are in 15 to 24 years age group, are most vulnerable group to STDs. Most vulnerable group of STDs 15 to 25. Early symptom, fluid discharge, pain, swelling. Okay, so these are some early symptoms of the STD. Late symptom, pelvic inflammation, ectopic pregnancies, right? So these are other symptoms of the STDs, late symptoms of the STDs. Infertility. Infertility means, right, unable to produce, the couple unable to produce children in spite of even unprotected sexual course since 2 years. Even after 2 years of unprotected sexual cohabitation, the couples are without children, right? So right, to overcome this problem, we should assist them with a special reproductive technology, or that is called assisted reproductive technologies, right? So we could assist the children, we could assist the couple to have a children with the help of the assisted reproductive technologies. Infertility means, in spite of unprotected sexual copulation even after 2 years, the couples are without children. So then we could assist them to have a children by following assisted reproductive technologies. So these assisted reproductive technologies divided into two types. One, in vitro fertilization, in vitro fertilization, and embryo transfer technology. Embryo transfer technology, ET, and in vivo fertilization. Generally, in vitro fertilization, in vivo fertilization, very important. Here, this is the area you will get question from this area. Some difficulty level question they will ask you from this area. In vitro fertilization, embryo transfer technology. It is commonly known as test tube baby. Commonly known as test tube baby. Here we collect sperm from husband, or donor, right? And we also collect ova from wife or donor, healthy donor. Okay, so now we are assisting to the couple, those are childless even after 2 years of unprotected sexual copulation. So we collected sperm from the donor and we collected egg from the donor, maybe wife or from husband, right? Now this fertilization of this egg and ovum occur under simulation condition. Simulation condition means similar condition. Generally fertilization takes place in ampulla region. What are the conditions that present in naturally in ampulla region? That conditions we are creating in the laboratory, that is called simulation. Simulation means repeating. Same condition we are creating in the laboratory, right? So now we are inducing these zygote, this sperm and egg undergo, we are inducing them to form zygote. This is the zygote. Now these zygotes will develop into 8 cell stage. 8 cell stage. In 8 cell stage, that means marula stage. 8 to 16 cell stage, marula. Marula that developing in the isthmus. So these 8 cell stage incorporated into isthmus, that is fallopian tube. Now you can see in vitro fertilization. The fertilization taking place outside the body in a special condition. That condition simulation similar to the uterus, fallopian, sorry, similar to the fallopian tube. And now these, after these, we will get a zygote. Now we are inducing that the zygote to undergo division, and it is producing 8 cell stage. Now these 8 cell stage zygote we are inserting into the fallopian tube. That is called zipped zygote intra fallopian transfer. Zygote intra fallopian transfer, right? When again you are inducing this zygote to undergo division, and it will produce 32 cell stage. The 32 cell stage embryo or zygote we are inserting into uterus. We are inserting into uterus. So that is called intrauterine transfer. In 32 cell stage, from the laboratory we collecting that and inserting into uterine cavity. That is called intrauterine cavity. This is zygote intra fallopian transfer. Zygote intra fallopian transfer and intrauterine transfer comes under in vitro fertilization. Zipped and intrauterine transfer, intracytoplasmic sperm injection. These three ART comes under in vitro fertilization. Here fertilization is taking place outside the body in laboratory, right? So we are calling them in vitro fertilization. Sometimes the embryo which is formed in vivo technique, naturally that is formed in a woman, that egg also we are using to transfer into another female, those who cannot produce egg. Okay, these are the technique comes under in vitro. And you can see gift, gamete intra fallopian transfer and artificial insemination. These are comes under in vivo. In vivo means fertilization takes place within the ampulla region of the female body. Natural, this is artificial outside the body, inside the body. So in vivo example, gift and artificial insemination. In gift, suppose this is the lady, she cannot produce ova, but her endometrium lining is suitable for the development of the ova, and fallopian tube is also suitable for the fertilization. So then we collecting the egg from the donor or wife. We collecting the egg from the donor. We are sending it into fallopian tube. We are inserting into fallopian tube. Ova. Natural copulation will take place. Because of the natural copulation, the sperm will reach here and fertilization takes place. Now what happened here? Ova is a gamete. Gamete intra fallopian transfer. We transferred gamete into fallopian tube. Now the fertilization occurring inside the body. So gyptis comes under in vivo technique. But here after fertilization, she can endometrium line can support for the pregnancy. Okay, this is very, very important. Next, artificial insemination. Artificial insemination is a technique that can be done if a male partner is unable to inseminate, or when the sperm count are very low or very less. That time we are following artificial insemination. Now we are inseminating the semen or sperm into vagina. Through the vagina they are migrating to the fallopian tube. So it comes under in vivo technique. In vivo technique, right? So artificial insemination, intrauterine insemination. So artificially we are injecting the sperm into uterus cavity. So that is called intrauterine insemination. Naturally it is moving to the ampulla region where fertilization takes place. Okay, so these are the in vitro techniques, in vivo techniques. Question mostly they ask you 8 cell stage transported into fallopian tube, that is gypt. More than 8 cell stage, uterus, intrauterine cavity, gypt, IUT and intracytoplasmic injection. Directly we are injecting sperm into egg or ovum, cytoplasmic injection. That 3 comes under in vitro fertilization, artificial insemination, and gift comes under in vivo fertilization. Okay, so I hope you understood this concept, right? So we covered most of the topics that we can expect question from those topics we covered. I think this video will helpful for you. Thank you. Okay.